So as Kat said, I'm currently based with the CSIRO in Canberra, looking more into reptiles, and they will just be like this tiny, tiny little side note at the very end of my talk, because the main focus of my talk today is what I've done over the last decade in the bottlenose dolphins in Shark Bay in Western Australia, as well as during my postdoc at UNSW. So this talk today is all about the glue that binds and my long, long journey learning a lot about friendships and also a little bit about genetics. So when I started my journey into biology, I was really fascinated by humans and our incredible level of cooperation. So what always fascinates me every single day is how cooperation just makes everything possible that we're doing. So every time, for example, we go into an airplane, we put our lives willingly in the hands of strangers. We trust not only that the pilot brings us safely from our starting point to our destination, but we also trust that the pilot and everyone else wants to go to the same destination that we're starting from, to the destination that we want to go to. Additionally, in the current times, none of us creates their own food. So we also trust strangers to produce sufficient amount of food, but also to produce that food safely. In this very moment now, I am also trusting on modern day technology and all of the smart people having written that software that to make it possible that I'm delivering this talk from the other side of an ocean to you in a completely different place. But also when we're thinking about how we're doing research in the current day and age, none of us has built their own hardware and written all of the software and code that they're doing. So even doing research in this current day and age is all about cooperation. Just about here, I have this tiny snippet of my son and my husband that just summarizes all of this cooperation pretty nicely. We are in Canberra at the Hot Air Balloon Show, where you see lots of technology, where multiple people cooperated to build hot air balloons to make humans fly. My son is sharing a croissant that neither my husband nor I made or harvested ourselves. And we're all happy in this huge assemblage of people. So that's a little bit about the cooperation and the amazing cooperation that we see in humans. But on the other hand, cooperation is also what, to me, makes all of life possible. We see cooperation within cells, between organelles. We see cooperation between cells in multicellular organisms. And we also see cooperation either within or between individuals of the same species. So cooperation is not only something that we observe on this really, really high level of complexity in humans, but it is truly the foundation of life. When I was starting my journey in biology, I came to realize this, but was really, really interesting to me was this high level of complexity and how we reach that. So on all of these pictures that we see here, it's not always just cooperation between two individuals, but it's this multi-level cooperation where it's not just a few individuals having a shared goal, but there's multiple layers of people that do cooperate that make everything possible. So this all sounds very complicated. I like to sometimes bring things back to something very simple. Let's just go back to the simplicity of a soccer field. So there you also have your various team. There's a few players that have the ball. You have your strikers, you have your defenders that in all aspects are kind of nested or independent little unique teams that are part of a larger team with a shared goal. So for me, this is kind of what sums up multi-level cooperation is people do different things. Sometimes they do it together, but they're all part of this larger cohort having a shared goal. So this is, again, a summary of what I have just talked about. And what I was really interested in is how did this evolve? How can we explain this? Really simple. We can explain the picture on the very right side with kin selection. kin selection is something that we observe a lot in nature. It just means that we take care of each other because we have a similar or shared set of genes. However, we as humans, we cooperate every single day with complete strangers. So how can we explain this? I was then thinking, as a young biologist, surely other primates must have evolved something similar. Let's just look at primates and then figure out how we share these traits and look at how other species have evolved similar systems. What I then learned was that I actually had to not look at primates, but that we observe a really, really remarkably similar system of complex cooperation, not in other primates, but in a population of bottlenose dolphins in Shark Bay, Western Australia. So this is what brought me, after my degree in evolutionary anthropology, to Western Australia. I will explain this population now in a little bit more detail. It's an incredible population of bottlenose dolphins that we have studied there over the last 40 years. It's not only that these dolphins there use culture in the forms of either sponges or shells that they use to forage, but they also have this unique system where both males and females form long-lasting social bonds. This is because neither males nor females do disperse from their natal area, so both sexes have the opportunity to form persisting social bonds. What is really interesting is the amount of cooperation that we observe in the males there. And this is very unique because in most animal species, we do not observe male cooperation. This is because in most instances, males have to compete with each other. So they are not naturally inclined to cooperate. And on the other hand, it's also, at least in mammals, most often the case that males do disperse from their natal area. So they don't also have the opportunity to form social bonds. So in Shark Bay, what drives, or what we think drives, cooperation among males is that there is no paternal care in the dolphin system, and the fact that females only give birth about every 4 years to a single calf. So that means that at every single point in time, there's a lot more males ready to mate compared to females, which results in an incredibly high reproductive operational sex ratio. So what we have in Shark Bay is what we call multi-level alliances, where multiple males cooperate for access to females. So we have what is about between 4 to 14 males that form second-order alliances, which are the core social unit of these males. Within second-order alliances, 2 to 3 males form what we refer to as first-order alliances, and these first-order alliances, they serve to for a few males, 2 to 3, to cooperate with each other and to coerce females into matings, which we refer to as consortships. The second-order alliances are the core social unit because they can last up to multiple decades. So the study site, or since we first figured out the whole alliance system, which was in the early 90s, some of these alliances still do exist, so they are incredibly long-lived. And the only thing that can change these alliances is if a few members either disappear or they die. The first-order alliances, they are always formed within these second-order alliances, but in contrast to the second-order alliances, they are less stable. Within first-order alliances, all males are presumed to mate with the consorted females, so there's no clear dominance hierarchy within these alliances. So this is also shown here, and just have a look at the top male here. So he consorts the female in gray with 2 of his second-order alliance members, but then the next time they consort another female, this time depicted in blue, he chooses 2 other members of his second-order alliance to form a first-order alliance with. The second-order alliances, they also serve to defend females in case other males from an opposing alliance come and try to steal their females, as they are an incredibly scarce resource. And females are dependent on both alliance levels. So in case a first order alliance level comes a little bit under pressure and might have their female taken away by an opposing alliance, they can ask their buddies from their second order alliance come to aid. But at the same time, the opposing alliance might do exactly the same and say, we really want to steal this female. Please, can you come help us? Which then again brings me to this nice example that it looks really similar sometimes what we do see on a soccer field. So this also, with this picture or with this explanation, I'm also hoping to convince you that this cooperation level that we do observe in the dolphin system is of incredibly complex nature. Now, a little bit more background on our study population. It is one of the longest monitored marine mammal populations that we have. Behavioral data has been collected systematically since 1984, so for 40 years. And genetic samples have been collected since 1997. Behavioral data is collected in the form of 5-minute ad libitum surveys, and we do recognize the individual dolphins based on modifications in their dorsal fin. To date, there's more than 1,000 individuals in our photo catalog that we can individually distinguish from each other, and it happens very rarely that we encounter an individual that is not existent in our photo catalog, so in a completely new animal. Then to come a little bit more into detail of how we infer their social networks, because as I said, this talk is all about social bonds and friendships in animals. So what we do in dolphins is that we do infer their social bond strength based on associations. And the association index is calculated as the simple ratio index, which is a value that is being calculated on how often we see two individuals together versus separate. So an individual that is always seen in association with another one, or a diet that is always seen together and never seen apart, has a simple ratio index of 1, and two individuals that have never been seen in association have a value of 0. So on the other hand, if two individuals are seen approximately 50% of the time together, they have a simple ratio or an SRI of 0.5. So at the very start of my PhD in 2014, we were curious about who forms these alliances. We had the hypothesis that it would be a quite simple idea of simply proving that alliances are formed among kin. This comes from the fact that most acts of cooperation and most alliances that we observe in primates and among mammals are generally observed between kin. So we thought in male dolphins, they share matings, so it would make sense to share matings with your kin because there's only one offspring per consortships, it's multiple males. So it would biologically make sense to go into these consortships with your relative, because even if you do not succeed in siring an offspring yourself, but your brother does, you still pass on your genes indirectly through that relative. I then created genetic data using a DD-RAD protocol, where we gained an overall amount of 9,991 SNPs to reliably estimate pairwise relatedness. I was then first looking at the second order alliance level to investigate whether second order alliance members are more related than we would expect by chance. And for this, I also was looking at the second order alliance choice. So I was looking not only at which males did actually end up in these second order alliances together, but also which males would have been around as potential partners, but were not chosen. For this, I looked at a total of focal males, of which I had adolescent and adulthood data available for. So the data that we had available during their adolescence allowed me to retrospectively identify their cohort of potential but non-chosen alliance partners. And then the same in adulthood. So for these males, I was then also available to look or able to look at their actual chosen second order alliance members. The way I identified the potential second order partners in a male's adolescence was for each focal male, I looked at who was around at the time, but I excluded all individuals that were too young at that point in time to be chosen as second order alliance member. I also excluded all individuals that disappeared while a male transitioned from adolescence into adulthood. And I also excluded all of the males outside of a male's home range because a male can only choose other males as second order alliance partners if it knows of their existence. On average, each focal male had a pool of 54 individuals that it could have chosen as second order alliance members during adolescence. And then to infer the actual chosen allies in adulthood, I used the simple ratio index because it has been shown that a value of over 0.2 usually clusters individual males into their respective second order alliances. So I use this as a cutoff, but to also use a functional behavior to classify males into their second order alliances. I also looked at who did they consort with, so that I also had a functional behavior to give me more confidence in my definition of who was chosen as a second order alliance member. There, on average, each male had 10 actual chosen second order alliance members once it was adult. I then looked at individual level differences between each focal male's chosen versus non-chosen males with regard to social bond strength in adolescence, age difference, as well as pairwise relatedness. And what I have found that was that relatedness did not influence partner choice at all, but the interaction between age difference and social bond strength in adolescence did. So interactions are always a little bit tricky. So I have made this graph and I'll walk you through it to help you understand it a little bit more detail in how this interaction needs to be interpreted. So overall, males are more likely to end up in the same second order alliance member if they had a really strong social bond during adolescence, but males that were older or of the same age as the focal individual were chosen if they had a little bit of a lower social bond strength compared to individuals that were younger compared to the focal. So it seems that in order for a younger male to be chosen as second order alliance member, the social bond needed to be stronger. So this was at that point in time a little bit disappointing to me because one always likes to find something significant. And I was thinking this could be a nice example of kin selection, which it wasn't. So I had another look at the level of the first order alliances, where I was thinking it would make even more sense if relatives are chosen on that alliance level, simply because males do engage and in consortships jointly and they mate within their first order alliances. So even if you don't choose relatives as second order alliance partners, it would make sense to choose them as first order allies because you do share your matings within first order alliances. Also there, I did a very similar analysis where I included individual level differences, this time between preferred and avoided first order partners on the first order alliance level in regard to social bond strength, age difference, and relatedness. But also there, only social bond strength was a significant predictor of first order alliance partner choice, while age difference and relatedness was not. So the expectation that I had at the very beginning of my PhD could clearly not be supported, as this was not an example of kin selection at all. So why could it be? Why do males not choose kin? It could be an example that kin can simply not be identified reliably. But it could also be, and I think that might be more likely, is that kin does not have preferred traits. Cooperation is in consortships is a really risky and complex task. You need to know who your friends are, who your enemies are. You need to know which females do you want to consort, and you also have to jointly defend that valuable resource. So it could be that in these kinds of cooperation that you do not choose your relative, but you actually prefer to choose someone who is reliable and maybe more competent compared to your relative, because your relative might not be the most competent partner in this task. So your relative might not be as strong or coordinated in his activities as a specific male. In that system, it is also very likely that demographic constraints limit the kin availability. So there is no dominant male of a given year that sires a cohort of siblings, which means that there might not be a large enough number of kin around to form these larger alliances. This would be very similar to what is observed in chimpanzees and Assamese macaques, where cooperation is observed among non-kin male because there's not enough kin available. But what I also did, I looked at the kin availability, and dolphins also do not even choose or prefer relatives as alliance partners when they are available. So overall, having found this strong signal of social bonds, it seems that social relationships are more valuable compared to relatedness. And I was then also wondering whether there is an additional adaptive value of these social bonds, so that they are not just serve as a predictor for alliance formation, but if there is also another value that is inherent to these social bonds. If we look at adaptive value, I like to come back to the concept of direct fitness and the number of offspring, as this is always a very good indicator of how successful an individual is in passing on its genes to the next generation. So in most animals, and especially among males, we observe a large variation of reproductive success. And the more offspring an individual sires, usually the higher its fitness is. And as I said, these differences in reproductive success, they are particularly pronounced in males, because males are constrained in their reproductive success by the access to fertile females, versus females are more constrained by the access to safety and resources. Over the past 100 years, there have been multiple variables identified that do influence reproductive success. One of them is age. In certain species, older individuals sire much more offspring compared to younger ones, or vice versa. Size, health, and strength and social status are also all very important predictors in a lot of species, as individuals that are stronger are often more likely to have a high rank and therefore be able to monopolize more females. Similarly, habitat size can be a very strong predictor, as individuals that have larger habitats are more likely to encounter or have more fertile females in their habitat or in their range. And very lastly, social bonds have more recently been added to this list as also being determinants of reproductive success. The influence of social bonds on females has recently been found to positively affect fitness on various levels, such as offspring survival and also offspring longevity. But this research has mainly been carried out in females. This has also to do with the fact that in most species, females remain in their natal area. So females do have the opportunity to form social bonds. But at the same time, this also means that it's much easier to obtain long-term data of females, because if you want to have data on a single male in a species where the males disperse, it's much harder to follow that male where it goes. So social network data is often much more easier to produce in females. However, in a few cases, there have been benefits found of male-male social bonds, such in Assamese macaques and in chimpanzees, where males that have stronger or more social bonds have been found to to be more likely to attain a top dominant rank and therefore to sire more offspring. Overall, all of this research has just been highlighting the fact that it's not just important to look at a social status, but also to consider an individual social environment when we are interested in investigating what could contribute to its fitness. So I found the male dolphins, again, a really interesting case study where we can look at how do social bonds contribute to fitness in males. I inferred the reproductive success in form of using classic microsatellite data, and I had data on 85 males that were members of 10 different second order alliances. I looked at 3 different social variables, the first one being first order alliance stability, which is a variable that informs about how often a male chooses the same set of partners from its second order alliance to form a first order alliance with. So this can be males that consistently choose the same set of males to form a first order alliance with, while some other males might choose them more what appears random. Then node strength and the coefficient of variation in social bond strength, which I will explain in more detail on the next slide. And as non-social variables, I looked at average age difference to second order alliance members and whether that influences a male's reproductive success, as it could be that a male that is older and maybe stronger and more experienced could sire more offspring compared to its younger partners. And I also looked at an individual's home range size. So when we look at how we calculate node strength, or often referred to as weighted degree centrality, this is I looked at each focal male, and then I looked at all of its simple ratio indices to its second order alliance members, and then the sum of them gives a male's overall node strength. So this is kind of a very simple measure of a male's overall social integration to its second order alliance members. What it doesn't do is, as in all of these 3 examples, all of the males have the same node strength, but the distribution of their social bonds is quite different. So the first male has equally strong social bonds to all of its second order alliance members, whereas the second one has a few what seems preferred versus avoided partners, and the third male has one really strong social bond and then lots of weaker ones. So this is where the coefficient of variation in social bond strength comes into play, as this provides us with an idea of the distribution of a male social bond within its second order alliance partner, or its second order alliance. So when I then ran a linear mixed model using all of these variables, I found that none of the non-social variables was significant, but 2 out of the 3 social variables, namely node strength and the coefficient of variation in social bond strength. Node strength positively predicted the number of paternities. So a male that is overall more socially integrated has a higher number of paternities compared to a male that is socially less integrated in its second order alliance. And then the coefficient of variation, I found a negative correlation, which means that males that overall have more homogeneous social bonds to their second order alliance members have sire a larger number of offspring compared to males that have a strong preference for only a subset of second order alliance partners. So this was a really nice final chapter for my PhD thesis, to not only show that social bonds drive alliance formation, but that there's also an adaptive value of these social bonds in the form that they do increase paternity success. And this is also complementing the female side very nicely, where it has been shown that social bonds among females do increase calf survival. So in this population, it truly seems to be the case that the more socially integrated you are, the more successful you can be. I was then at the very end of my PhD, and I felt like there was still more to explore because, as I said at the very start of my PhD, we were thinking this is all kin selection, and then it turned out that it wasn't. So I was wondering whether there were even more benefits of social bonds. I also wrote the very last bit of my PhD in lockdown. And that's when it became clearer and clearer that in humans, social isolation was not a good thing for our health. There were more and more studies coming to light about how the lack of social bonds and social isolation is affecting our mental health. At the same time, it was also more and more research done, not just on our mental health, which is probably just what we focused on, but it became clear that social bonds do not only affect our mental health, but actually our overall health and longevity. Now, this is not completely new when we look at research that has been done in social animals, where it has been known for a little bit longer that social bonds affect lifespan and survival. So in social animals, individuals that are socially more integrated, they often have a longer lifespan or better rates of survival compared to individuals that are more socially isolated. This is what I was then thinking of investigating in the dolphin system. So I was thinking if social bonds influence health and survival in social animals, and we have this incredibly social population at our hands, we should have a study on this. Unfortunately, survival is incredibly difficult to measure in a long-lived animal. I had a look at the data, and I think in our overall records, I found about 5 individuals with complete life history data where we knew when an individual was born until when it was died. And n equals 5 was simply not a sufficient enough number to base a whole postdoc on. I was then thinking, well, instead of just looking at life history data and lifespan data, I just measure the pace of aging. So a physiological measure of age, so biological age. Sounds much more complicated than just to look at life history tables, but it is thankfully a little bit, it's not as complicated as it first seems. So first of all, I thought this idea is crazy. But I think I had the right idea at the right time because that was also around the time when epigenetic clocks started to be a little bit more established as biological age estimators. Epigenetic clocks are based on CpG sites that gain or lose methylation in a highly predictable manner over a lifespan. Methylation are these little chemical tags that you find on your genome, and they influence whether a gene is expressed or whether it is not expressed. So it doesn't change the genetic sequence itself, but the way genes are expressed. Epigenetic clocks, or these regression models that we find using these CpG sites, they have been found to be the most accurate biomarkers of chronological age. But what is really interesting is that it appears that they are also a marker of our biological age. Individuals whose epigenetic age is older than their chronological age are often the ones that are in not as great health. And this opposes individual whose epigenetic age is younger compared to their chronological age, who are often appearing in very good physiological health. So it is not surprising that smoking and alcohol use has been shown to increase epigenetic age in comparison to chronological age. Also, male sex is related to an increase in epigenetic age, and this probably has to do with the spans compared to females. On the other hand, unsurprisingly, if you want to lower your biological age, it's the normal things that every physician usually recommends you to do, which is engage in physical activity, eat a healthy diet, but at the same time, and what's really exciting is have social bonds. So I always say, maybe it's better for your health to skip the run and go for a beer with a friend than to just go for the run alone. So you can pick and choose a little bit of how you interpret this. But this was really exciting for me to see that there are social bonds and that they can actually have an influence on your epigenetic and therefore your biological age. So how do we build these epigenetic clocks? Because as you might know, there are a lot more CpG sites in your genome than we probably have dolphins in the entire ocean. So we use a method that is called elastic net regression. And elastic net regression allows to have a much larger number of predictors than a number of individuals included. So your predictor, it's very similar to a linear regression. Your predictor here is chronological age. Then we include all of the CpG sites that we have methylation data for. And then the elastic net regression, which is the machine learning algorithm, looks at which CpG sites do actually correlate with age and nicely sums them up into a model. So I know that a lot of you at the computational facility would probably be really, really interested in that part. And also a lot of you are probably more experienced in that part. So I put it in here. If you are really interested in learning more about how this works, then please speak to your colleagues out there. Feel also free to speak to me and to maybe hook me up with one person that knows a lot about machine learning in your department, because I'm more than happy to learn more about it. So we then use to validate these models a method that is called cross-validation, which means that during the model generation, you split your data set into a training, a validation, and a test data set. And you do this a number of times. So your training set is always the one set that you keep, and then you validate it on like a smaller subset, and then you look how your model performs on the data that you've purposefully left out, and you look at how does your model actually perform. This I left in here because what I used in my model was a leave one individual out cross-validation, which means that every time I trained the model on all samples that I had, apart from all of the samples that I had of a single individual, and then applied the model to the left out individual. And this allowed me to avoid that I trained and tested the model on data that I had collected on a single individual, and therefore to just make sure that my data is as independent as possible. So the epigenetic clock that I ended up calibrating for the shark bay bottlenose dolphins, as I said, I was using a leave one individual out cross-validation. It performed quite nicely, having an R-square of 0.86 and using an overall of 90 samples that were collected from 68 individuals. Then to look at whether social bonds affect the pace of aging, so whether social bonds are correlated with epigenetic age, I built a linear mixed model in which I included epigenetic age, and then I looked at whether node strength, sex, and then correcting for chronological age, what I could find in there, whether I could find any influence of node strength on epigenetic age. And indeed, this is what I found. I found that individuals who had a strong or stronger or higher node strength had lower epigenetic ages, which means that individuals who are more socially integrated in their social networks are epigenetically younger compared to individuals that are less socially integrated. I also found a tiny effect of sex, and that also is exactly in the order that we would expect it, in that males are estimated to be epigenetically older compared to females of the same age. And this fits nicely with the mammalian pattern, where most of the time males do have a shorter lifespan compared to females. overall, this was a really nice study to see that the pattern that was observed in humans matches the one that I have observed in the dolphins. And while this is really exciting, I also do think that this is the pattern that we would expect for any social species, simply because social species have evolved to thrive in a social environment. And as soon as this social environment is no longer in place, as stress levels increase, and therefore I could also imagine that overall health and therefore biological age suffers. I would be very interested to see what is being found in other social species, and if this pattern that I have been finding in the dolphins and that was observed in humans holds up across social species. I unfortunately don't know anything about the causality, so I have no idea whether individuals that are biologically younger simply have more energy and are in better health to also invest energy into social bonds, or if it's actually the social bonds themselves that do lower epigenetic ages, such as I said, for example, via levels of having reduced level of stress or other pathways. I think this one here is a really nice study, then also again to just make a statement that these incredibly expensive and time-intensive long-term study sites, such as the one that we had over 40 years in Shark Bay, they can increase our understanding of fundamental biological processes such as aging and cooperation. So just to end it up, I said I would just end with a really, really brief note on where I am going next. So at the moment, I am calibrating an epigenetic clock in a non-mammal species, namely in the bearded dragon. And what I'm hoping there is to identify environmental variables that affect the aging process, as well as maybe even identifying some loci that are core conserved in the aging or affecting the aging process across vertebrates. Now, while this might seem that I'm no longer interested in cooperation, it actually is, I have started to also see aging as a factor of cooperation. As I said at the start, cooperation is something that we observed on all levels of biological organization. And I do think that what happens while we're getting older in cells is that they their genomes or their epigenomes don't cooperate at the same level anymore, which is then driving everything a little bit to not as good of an equilibrium as it was when we were younger. So I can say that I am finally back on land after an excursion into the ocean. And I think also after having done no primate work since my master's thesis, I can no longer really call myself a primatologist, but I am definitely very, very happy to be back researching a terrestrial species. Very lastly, just to make another statement about cooperation. I will be in New Zealand in early December attending the Australian Epigenetics Alliance conference. So if you would like to cooperate with me, form a social bond with me while I'm there, then please get in touch. And that is all I wanted to present to you. And I thank you all for listening, and also to Kat for contacting me and inviting me to be here today.